We're at session 3 of the workshop, and I would like to introduce the chair for this particular session, Dr. Agam Vora. He's the medical director of the Vora Clinic in Mumbai, formerly the assistant honorary consultant in charge and at the Department of Chest and Tuberculosis, the Dr. R.N. Cooper Mooney General Hospital in Mumbai. Is the assistant professor of the Department of Chest and Tuberculosis at the KJ Somaiya Medical College in Mumbai, the assistant editor, Journal of the Association of Physicians of India. He's on the editorial board of Chest India, the General Secretary of the Academy of Advanced Medical Education, the past president of the Geriatric Society of India, and the trustee for the Malad Medical Association. So it gives me great pleasure to hand you over to Dr. Agam Vora. Thank you.
Good afternoon. Thanks, Mr. Kim, for those nice words of introduction. And it's my proud privilege to be here in front of you today on this second and the last day of APACI Respiratory Workshop. Over the last 2 days, we've been talking about vaccination. The topic vaccination is very close to my heart. WHO says, and I endorse, vaccination is the only strategy to prevent a lot of vaccine-preventable disease. We have at least 25 life-threatening disease prevention possible due to this vaccination. I'm a strong believer of vaccination, and I vaccinate about 1,200 influenza vaccination every year from my clinic, and pneumococcal vaccinations I give from my clinic to COPD patients. I've been fortunate to be part of vaccination recommendation by Indian Medical Association, Association of Physicians of India, and also Geriatric Society of India. I'm very happy that this program is endorsed by API and ICS both, so that more and more delegates would be able to participate into this event and get benefited. With that, allow me to introduce the next session, the session which is going to be on COVID vaccination and superinfections. And for that, I have the excellent panel with me. The discussion is going to be moderated by Professor Robert Boyd. Professor Robert is the Director of Immunization Coalition in Australia and a Senior Professional Fellow at Children's Hospital, Westmead School of Child and Adolescent Health. He's done MBBS, MSc, MD, and is fellow of a couple of international organizations. On professional front, he graduated from University of Queensland in 1984 and trained in pediatrics at Royal Children's Hospital, Brisbane. He's the professor and head of Department of Child Health at the University of London during 1999 to 2005. He is recognized as an expert in COVID and influenza fields, supervising many studies addressing the burden of and prevention of respiratory viral disease in both children and adults. I present Professor Robert Boyd to you. Next, I would request Dr. Gary Grumman. Dr. Gary Grumman is the Director of Australian Immunization Coalition. He's a consultant at WHO Committee on COVID and Influenza, that is SAGE. He's a Director of Environmental Pathogens, Adjunct Professor, Associate Professor in the Department of Infectious Disease and Immunology at University of Sydney. His BSc and PhD, also holds a fellowship. His previous assignment with WHO were Technical Advisory Group of Global Action Plan for Influenza, review of IBTM database on influenza viruses and assorted influenza viruses. He's even the vaccines, PIP, review secretariat and consultant on switching from seasonal to pandemic vaccine production, review of biocontainment document. I invite Dr. Gary to join us. We have Dr. Paul Griffin here with us. Paul is Associate Professor of Medicine and Director of Infectious Disease at University of Queensland and Masters Hospital, South Brisbane, Australia. He's a Director of Australian Immunization Coalition, Principal Investigator investigator and medical director at Nucleus Network, Brisbane. He is MBBS and holds a couple of international fellowships. He's received fellowships in infectious disease from Royal Australian College of Physicians. He's received clinical microbiology from Royal College of Pathologists of Australia and from Australian College of Tropical Medicine. I invite Dr. Paul Griffin to join us. We have Dr. Sheldon Campbell. Sir is the professor of Department of Laboratory Medicine and Associate Chief for Laboratory Medicine at Connecticut Healthcare. He is PhD and the professional honors are recently 2015 to 19 he was member CDC Clinical Laboratories and Advisory Committee. Earlier to that he was Outstanding Speaker Award American College of Association of Clinical Chemistry, received that in 2015, and 2012 to 13 he chaired American Society of Microbiology, that is Clinical Microbiology Division. And we have Dr. Randeep Guleria here with us. Dr. Randeep Guleria is Padma Shri, MD and DM Pulmonary Medicine and Critical Care. Currently, he is the director of All India Institute of Medical Sciences at New Delhi. He is the first DM Pulmonary Medicine in our country. Recipient of several awards, innumerable awards. And the most prestigious one would be Padma Shri, that was conferred to him in 2015. BC Roy Award for Eminent Medical Person in 2014. Himachal Gaurav Puraskar, Swasth Bharat Samman, Legions in category of Doctor of the Decade Award. He's received award by Indian Medical Association, he's received award by all professional organizations in the country. He's delivered more than 8 prestigious national and international orations with more than 450 publications, with 50 chapters, 50 research grants, and more than 300 invited lectures. He was personal physician to Prime Minister of India, personal physician to many international and national dignitaries. He is the advisory board member of various international and national bodies. On personal note, extremely humble, down to earth, polite, soft spoken, very well composed. And in my 15 years of association with him, I've never seen him angry or I've never seen him upset. He always wears that signature smile on his face. With that, I present Dr. Randeep Guleria to you. Before we move on to the scientific session, I also have one announcement for the poll question. And the questions are 2. I want you to answer. Have you received your first COVID vaccination? Please put yes or no. And the next question would be, have you received your second dose of vaccination? Yes or no. Please put your answers. And by the time we receive your answers, I would request Professor Boyd to start the session. Over to you, sir. Sir, you're on mute.
Good morning and good afternoon, everybody around the world, in the Americas, in India, in Australia. It's a pleasure to be with you. I'm in a cold place near the city of Sydney in Australia, and it's a tremendous pleasure to work with this faculty. I'm very excited about this session, and I'm wondering how many of you have had your first and second COVID vaccines, and maybe someone in the United States has even had their third. I do hope that everyone has had at least their first, and seriously, this is a major, serious international problem, and I'm looking forward to the presentations and to the discussion we're going to have. Has anyone got results already on the survey, or shall we wait till later for that? I'm not hearing any results. No? Okay, well.
Any answer?
Yes, sir. We'll just put the graph on screen. Just give us a second.
Okay. Well, if someone's been writing in the chat, they've had both their doses, so that's fantastic. Clearly, there are different vaccines to be had as well, and some discussion about the different types of vaccine being used would also be instructive to all of us, because we have all different international experience and different experience with disease and with vaccines. Tremendous steps have been undertaken in India to produce locally and internationally millions and millions of doses. So here we go, starting to screen. Have you had your first? It looks like 100%. Well, that's what I would have expected, but that's great. And for the second, there looks like there's only one person who's yet to have their second. So that's not too bad, considering most vaccines provide high levels of protection, even from the first dose, from the third week after the first dose. That's fantastic. We seem to be preaching to the converted, but it's actually a much deeper issue that we're going to be discussing: the disease, its manifestations, its complications, and its prevention. So I'll stop speaking now. I'm going to take lots of notes, and when I'm asked again, I'll help to moderate discussion. Back to you. Okay, it's an international conundrum. Are we about to hand over to the first speaker?
Robert, it is a general discussion. There are no formal talks.
We've got a fantastic faculty. Can I invite Paul Griffin to open discussions, and then we can have some to and fro once Paul has spoken.
Thanks very much, Robert. Hopefully you can hear me okay. Look, it's certainly an honour to be part of such a prestigious faculty, and I'm very grateful for the invitation to speak. My understanding of the session is that we're going to be discussing, I think it's co-infection and COVID and the role of vaccination. So I guess to start in Australia, we're very fortunate to have had a very different experience to, I think, a lot of other people who are probably participating. And where I am is in one of the northeastern corners of the country in Brisbane. And we've actually had a very aggressive suppression strategy. So have been relatively spared, having relatively few cases and seeing very little of the impact that a number of other people have obviously endured personally with this virus. So in terms of my discussion today, a lot of it will be based on what I've read from abroad rather than seeing too much personally. My personal experience here is we've looked after perhaps hundreds of cases, with very few of them being critically unwell. I guess the other aspect to discuss in terms of our experience where I am is that our vaccination rate is perhaps a lot less than other people. So maybe we have the worst yet to come here. So we haven't seen a lot of co-infection and severe disease locally here, thanks to our aggressive suppression strategy that at least for now is holding things. But I'd certainly be interested to hear what other people may have experienced firsthand and be able to relate to that from what I've read about other people's experiences.
Thanks, Paul. I'm going to keep this moving fast. I want to move to Sheldon. I think you're on the east coast of North America, am I right? And what would you have? You must have a completely different experience to Australia. Tell us about it.
Entirely different. We were not very successful in suppression strategies, as most of you probably know. And our experience with vaccine and the outbreak has been somewhat still inexplicable. We began vaccinating people in late December and initially had a good uptake of the vaccine. We've since run into vaccine resistance, not by the organism, but by the people. And the initial rollout of the vaccine was accompanied by a fairly substantial drop in incidence. And over the course of the first 5 months of this year, with a marked decline in incidence, much before the weather turned, to make us think that other factors were intervening, climatic and epidemiological factors were intervening. But then with the advent of the Delta strain, despite higher vaccination rates in my state and in other parts of the country, the incidence of disease has risen again. We still have very little severe disease and remarkably low hospitalization rates in vaccinated persons, but we have quite a lot of breakthrough disease, just not severe disease, and undoubtedly significant transmission in our vaccinated population.
Sheldon, thank you. I'll keep moving fast. I'm actually a pediatrician, but I'm interested in the elderly as well. And the only death we've had in Australia in a child is a 15-year-old who died of a co-infection. It was pneumococcal meningitis. And I reviewed the literature from around the world, and there are many short case series and case reports of pneumococcal septicemia and meningitis in association with COVID, which is indeed interesting. What's the experience in India? Professor.
Thank you very much. And as far as India is concerned, our vaccination drive started in January of this year. We were hit then by the second wave, which was basically initially by the Alpha variant and then the Delta variant. And it led to a huge surge in the number of cases. We had almost 4 lakh cases a day for quite some time. And we are now seeing the tail of the second wave in states in the south. In Kerala, we are still having a significant number of cases. We're around on an average of 40,000 cases a day. But our vaccine drive has picked up since January. And I would say that we are able to now have a good vaccine strategy. 2-3 days ago, we were able to, in one day, vaccinate 10 million people on one day, which was a remarkable achievement. And we have now almost vaccinated 60 crore individuals. That is, our population is 1.2 billion. So we have actually given one dose at least to half the population, the adult population. So that is also remarkable. We are actually moving ahead in terms of vaccination. The interesting point is that, as has been mentioned, India has been moving aggressively. Initially, India was the hub for vaccine production. We used to produce 60% of the global vaccines when it came to pediatrics and the pregnant women. We have now also started doing R&D in vaccine, and India has now its own vaccine, which is the attenuated, the whole virus attenuated vaccine by Bharat Biotech. Serum Institute is, of course, making the AstraZeneca vaccine. Another company, Zydus, has got approval for a DNA vaccine, which is the first in the world. It uses a DNA platform, ZyCoV-D, and that has also been approved for children above the age of 12 years. There's also a vaccine being made by Biologically, which is an mRNA vaccine, which can be kept at 2 to 8 degrees centigrade. So there is a lot of ongoing research, which is being supported by the Department of Biotechnology, Government of India, in terms of the funding that is happening. And as has been said, after vaccination, we are seeing less severe disease, but there is breakthrough infection, which is being reported, and it's a cause of concern. India also set up the INSAGOG, which is the surveillance network for the whole genome sequencing to look at the variants. And although the dominant variant currently is the Delta variant, there are now pockets where we're also seeing the Delta plus variant. And that lineage is basically AY4 and AY12, which is being reported. The data currently doesn't suggest that this variant is leading to more severe disease or leading to more infection. But I think this is one variant, the lineage, the Delta plus lineage, that we are monitoring very, very closely. And that could be a cause of concern, both in terms of breakthrough infections and the immune escape mechanism which the virus may develop. So I'll stop here.
Professor, I've got a quick question. There's a great Indian word called lakh, and does that mean 100,000? Can you just educate me for a moment? When you're talking about large numbers, you use a word called lakh, L-A-K, is that right?
Yes. So when we're looking at our population, we say that when we talk of, it's, you could say 100,000, let's say.
Yeah, I thought so. Thank you. Now, tell me this, and I'll go back to you first, Professor. In Australia, we had a record immunisation uptake against influenza in 2020. We actually achieved something like 80% of our population vaccinated against influenza, the best we'd ever done. It was done because many people were fearful of COVID, but they had no vaccine. So they had influenza vaccine instead in the hope that they could prevent a co-infection. Has there actually been much in the way of co-infection between influenza and COVID-19 in India?
So there have been some case reports, but it's not been seen that much. In India, except for what we see in the northern state, we have our surveillance data suggests that we have two peaks of influenza, one during the monsoon months, and currently we're going through an influenza outbreak in most parts of India. That is causing a lot of concern because the clinical manifestation is the same. It's a flu-like syndrome for COVID and for influenza per se. So we are seeing a number of individuals who have a flu-like syndrome, but they're all turning out to be influenza. In pediatricians, there's also RSV being reported, and COVID is not being reported that much. But that is happening, and we see this every year that during our monsoon months and then during the winter months, we have two peaks as far as influenza is concerned, unlike what we see in the northern hemisphere and the southern hemisphere. As far as vaccination is concerned, it has helped to drive influenza vaccine, but our uptake for adult immunization has been low, unlike in the pediatric age group, and I'm sure this will be discussed subsequently, but that has gradually picked up, especially for pneumococcal and influenza vaccine. Influenza vaccine has picked up. There is a lot of pressure for it to be made into some sort of guidelines, and as was mentioned by Dr. Agamvora, that many of the associations in India have actually pushed aggressively for adult influenza vaccination for the high-risk groups.
So I'll move to Gary Groman now back in Australia, who's done a lot of work with the WHO on influenza and also has been reviewing COVID. And I wonder if you'd like to talk to us about the interactions and also the vaccination and the registration and the sort of the safety aspects as well, from your point of view as an advisor to the WHO.
Sure. Thanks, Robert, and thank you, everyone, and to the organisers for the invitation as well. And just a little bit of background, I did work for 17 years for the TGA, head of immunobiology for the regulation of vaccines. So I can speak a little bit to that if you wish. But certainly from a WHO point of view, you know, it's a great reminder that the problem is not just local, that it's a global problem. And it's so important that we try and get vaccines and good medical care to as many people as possible in as many countries as possible around the world. The thing about registration is actually fairly straightforward. It's been an extraordinary journey, of course, in the last 14, 15 months with these brand new vaccines, never been used before in humans of any number. Certainly not for this virus or a similar virus. And for it to get through and to be registered in such quick time and for regulators to be reasonably confident, at least about short to medium term safety, and for the very large trials to be done, 30, 40,000, depending on the study, has really been an extraordinary effort. And the result has been that we now have extraordinary weapons against any future pandemics because these kinds of vaccines can be made at speed. And while there's still some concern about long-term safety data, it was good to see the FDA approve Pfizer after reviewing the data again and give it a full approval and not just emergency use approval. Every other vaccine in the world is actually for emergency use or simply provisional because of this issue of a lack of long-term safety data. But nothing at the moment is expected. There's a few fears around and too much nonsense on social media, but we just have to go step by step, and so far there's no indication of any long-term issues. So I would expect these vaccines to be declared fairly safe, on the caveat that there's still a little bit unknown. One very long-term effects, if any, or some weird or wonderful surprise that might come through, but that seems to be unlikely now. We don't know really if the effect of the third dose, if that will present any issues immunologically with a third dose. And we don't have the full story yet on mix and match, although from an efficacy and effectiveness point of view, it does look very good. From a safety point of view, there are more adverse reactions, and that still is being studied. And of course, we don't know about our under 12 age cohort, and there's been a push around the world to vaccinate teenagers and young adults because they appear to be spreading the virus more quickly. But we have to do that with great caution. I think as a former regulator, I would be incredibly careful of vaccinating children if there's no great need to do so in terms of morbidity and mortality. So that's my message from a regulatory point of view. In terms of superinfection, I think most of us did expect that there may be some issues with influenza and other viruses, and in particular bacterial pneumonia, of course. And these studies I've reviewed and read would suggest that some 8 to 14% of people with COVID do get a superinfection of bacterial pneumonia. I haven't seen too many numbers with a co-infection with other viruses, and that doesn't surprise me as a virologist, because essentially the viruses are competing for the same host. And we see this, we've seen this before also with an H1 being knocked out by an H2 or being knocked out by an H3 and so on over the decades last century. So that doesn't surprise me. And we know from an immunological point of view, which is of course the other half of the story, it's not just all about viruses, disease and vaccines. We do know that there are various markers there that are very important, like CRP, C-reactive protein, and pre-sepsin, and PCT, IL-6, and so on. And these are very, very important in terms of a proper diagnosis of COVID-19, particularly in the absence of PCR, depending on which part of the world. So it's important now, I think, to study what is going on. And we have no cases in Australia to speak of, so we can't really study it here. But certainly data is slowly coming through via the US and the UK and other places that can be relied on. And we should get a good picture, I think, of the story on superinfection. Over to you, Robert.
Thanks, Gary. Sheldon, I'm going to fly around the world to you, then back to India, then back to Australia to Paul, so everyone can get ready for what I'm about to do. But first, you, Sheldon, there are two issues. With influenza a century ago, 50% or more of people seemed to have a deadly secondary bacterial pneumonia, and yet COVID appears to be less than 15%. You've raised the question in the chat, what do we do with steroids if we've got a secondary bacterial infection? Would you like to have a go at addressing your own question?
Oh, my expertise is a lot more in diagnostics than in therapeutics, so I'm not sure I'm a good person to ask for an answer to my own question. Well,
you give us your answer that you want to give, and then we'll go back to India and see what they think.
The pathogenesis of severe COVID is so driven by the immune response. And dexamethasone really has a profound survival effect in that population with severe COVID.
Yeah.
I think it'd be very hard to get critical care providers to say, well, if we suspect a bacterial infection, and they have to make the decision before it's confirmed often, we're going to not use the dexamethasone. So I think on a practical level, where you have to make the decision, you don't necessarily know whether there's a bacterial infection happening or not for a day or so. Things like procalcitonin may be biomarkers that are mildly helpful for making that decision, but we don't have the data yet.
Well, as a pediatrician and as someone who's worked in intensive care, I can see people throwing the kitchen sink, and that's antibiotics, right, dexamethasone, tocilizumab, IL-6 antagonist, and there's a few more coming. And we know that your former president may have been saved by Regeneron, a monoclonal antibody, and made that miracle recovery in 4 days. Now in Australia, we have two monoclonals, Regeneron and sotrovimab, and they're both looking very promising from the trials. But I'm going to pass to India.
But
is there the scope? Go
in
on,
our
Sean.
experience, the monoclonals are not very effective in patients who are already acutely ill, critically ill.
Our indication, we've just bought thousands of doses. We're going to use it early on in people at high risk before they're sick.
That's the way we use it now. You know, the one other thing I wanted to mention is you were going through talking about superinfection, is that we've seen quite unusual superinfections. In my facility, we reported a case of strongyloides, and there have been large case series with things like mucor, where the lymphocytopenia and the lympho suppression of this virus has seemed to produce opportunistic infections of that nature, as in addition to the bacterial pneumonias.
Yes, that's a really good point. Mucor, I've been reading from India, is incredibly prominent, and I've turned to our Indian professor and expert. Tell us about your experience and that of your colleagues in India.
Thank you. So again, what I'd like to say is we do see secondary bacterial infection. Of course, it was much more common during the Spanish flu, but then at that point in time, antibiotics were also not available. But currently we are seeing, and especially since many of us work in critical care, patients who are in the ICU, especially those who have been intubated or on a ventilator, ventilatory associated pneumonia is common. We would continue to give steroids, and there is data both in terms of sepsis, but also from the point of view of pneumonia, that steroids may be useful. And considering the immune response that COVID really has, one would actually not only give steroids, but if there is a need, give anti-IL-6 drugs also, like you mentioned, tocilizumab. Secondly, we're also seeing a lot of mucor, and in our hospital itself, we had almost more than 300 patients subsequently after COVID who were diagnosed to have rhinocerebral mucormycosis. We had a few patients who had pulmonary aspergillus infection also. And these are secondary fungal infections occurring because of two predominant things. One was that we have a lot of diabetic patients. We are at times called the diabetic capital of the world, and many individuals have poor control of the diabetes, and that predisposes to these opportunistic fungal infections. Secondly, because of this huge surge in the second wave, there was a lot of overuse of steroids, even in those who had mild illness. And this led to a huge excessive use of steroids, leading to a background of diabetes, leading to a higher chance of mucormycosis. So there are hospitals which have seen more than 400 cases of mucormycosis. Our surgeons, especially the ENT specialists, had a tough time because mucor, you have to operate and give antifungal. Antifungal alone don't work. And that is also true for pulmonary mucormycosis. We lost one patient, just the surgery was planned the next day, the evening before, she bled because of her massive hemoptysis, and we couldn't save her. So mucor, I think you have to treat it, the surgical part has to be treated like an emergency. And our surgeons still today are still working and collaborating with the neurosurgeons, the dental people. It's a multidisciplinary research as far as rhinocerebral mucormycosis is concerned. The other interesting thing that we've seen, and this is a few cases that we've seen, and here again there's a challenge, is that we also have vector-borne disease. And in the monsoon months, we see a lot of dengue infection. And dengue is an infection which also causes thrombocytopenia. So you have a patient who actually you are treating with anticoagulants, and you're trying to address issues of a hypercoagulable state. And then suddenly this patient gets a vector-borne disease like dengue, and his platelets come down to 20,000, 30,000. And then you have this whole issue, what should you do? Do you continue anticoagulants knowing that COVID can lead to a pulmonary thromboembolism or hypercoagulable state? There is a feature of endothelitis in it. But the dengue itself has caused thrombocytopenia. So we've struggled very often with these patients and trying to sort of walk a fine line. So that type of superinfection also we are seeing in tropical countries. And I thought I'd just mention that because it's been challenging when these infections happen as far as our country is concerned.
If I may add to it, in probably next few years are going to be very interesting. It's going to be very learning phase, because I fear about tuberculosis as well. Luckily, with mass social distancing, etc., those tuberculosis cases are not coming in front. But with overuse of these biologics and overuse of these steroids, I fear next 4 or 5 years, we would be seeing a lot of tuberculosis that probably we could not diagnose at this time, or probably there was delayed reporting because the entire healthcare is busy in looking after COVID patients. They're busy in diagnosing COVID patients. They're busy in all the beds, etc. Everything is occupied by COVID patients. So this tuberculosis, I have my own fears about it. Thank you.
I think your fears are well founded. The co-infection is something that we're not recognizing as well as we might, but it's astounding the fungal superinfection that we're seeing in such a large number of people. Now, I know that Indian people in the United Kingdom are at increased risk of COVID. I haven't seen much in the way of secondary fungal infection in UK reports, but Professor, could you speak to that, the diaspora of Indians in North America or Europe?
So that's correct, that the number of fungal infections being reported from this part of the world, that is Southeast Asia, is much more than what is being reported even among the Indian diaspora in the Western world, whether it's UK or the US. But I think it's also driven, like I mentioned, that in these countries, you cannot really get steroids, let's say, off the counter. You have, and there is a strict limitation on the use of steroids in mild disease. Whereas in our country, you could buy steroids or any antibiotics from a chemist over the counter. And this led to a lot of misuse of steroids because of a panic reaction which happened because of the surge of the second wave. And that actually, I think, is one of the cause with the background of diabetes, which led to this huge cases of mucormycosis. And we actually moved from our hospital beds being full. We had more than 1,000 patients admitted with COVID. And when COVID came down, we had to create mucor wards because we had now a huge number of patients who were coming with mucormycosis, and they obviously are going to stay for a long time because you start them on liposomal amphotericin B, subsequently switch them over to posaconazole, but that takes a long time, and the hospital admissions is much longer than what you would get otherwise. And that created, again, a newer sort of strategies we had to develop, going from COVID to mucor to non-COVID care.
Well, it's an astounding challenge. I'd like to ask Paul if he'd like to talk about the sequence of treatments and how we can get ahead of these secondary complications, and when is the right time and the wrong time to use all the different treatments. That's a big task for you, Paul.
Well, I guess the simple thing to say there is it's evolving so rapidly, and it's going to be very different depending on where you are and what you have access to. I mean, to go as upstream as possible, obviously vaccination is our best way of addressing that by reducing the burden of infection. In this country, certainly we do use steroids when people particularly have an oxygen requirement and more in that inflammatory phase. And tocilizumab is something that we've used quite extensively, but have fairly limited supply now. I think that the monoclonal antibodies, sotrovimab particularly, is looking really promising, as has been said, for stopping progression to more severe disease in those people who are very early on, so not yet having any significant manifestations. And that's certainly something that we're looking at how best to utilize. We have 7,700 doses in this country, but are still grappling with how to get that to people that are positive as early enough that it's effective, but while still knowing who's going to derive the significant benefit from doing that. So we're obviously still at a fairly early phase in terms of treatment, but there's a lot of other things coming through, and this will obviously continue to evolve rapidly.
It's a challenge. We've got some people on the line who'd like to ask questions, I think. So just write your question in the chat and I'll find it and say it. I want to turn back to Sheldon to see where he's up to with the diagnostic aspects and other elements that he'd like to address before I then open it out a bit more widely to the chat. So do start writing in the chat, and over to Sheldon for his brief comments.
I mean, I think diagnostics early on was a limiting resource, at least in the United States, where we didn't control the initial infection well. We've gone through a phase in the spring when disease incidence was down, when we actually had an oversupply of diagnostic capacity. And now with the Delta variant and the increase in cases again, we're back to, in many parts of the country, having diagnostics as a limiting factor. It's been quite a roller coaster ride for us. In terms of interaction with new strains and vaccines, the vaccines have not been an issue for diagnostics. And so far, we have not seen diagnostic escape strains as we have for a couple of other cases and malaria for rapid diagnostics tests and things. And I don't necessarily anticipate it. We're frankly not good enough at contact tracing and prevention measures on the basis of diagnostics that there's that much selective pressure on the virus from diagnostics. Almost all the approved tests in the US, at least for molecular tests, are dual target tests. And one or two of them have lost one target or another to different strains, but none of them have lost both targets. And the companies are staying ahead of that. So the diagnostics are capacity is an issue, but right now the biology we seem to be keeping up with.
Okay, thank you, Gary. There's been a bit of discussion in the chat about the fact that influenza in the last year or two has hardly been seen. There's comments that from Professor Galleria that we may have influenza in the south, but not the north of India. In Australia, there doesn't seem to be any at all. What are your comments? You've been looking internationally at gizzard, at COVID, at flu. What do you think? You're muted, Gary. You're muted.
Yeah, apologies for that.
There you go.
Yeah. Well, as we all know, the data shows pretty clearly that flu has, I wouldn't say disappeared, but it's certainly, there are certainly very, very few reports compared to previous years, including deaths and pediatric cases. So I've seen that around the world. As I said before, I think it's a phenomenon of the fact that we have two viruses potentially circulating that affect the one system. So essentially flu runs out of hosts because COVID is a lot more infectious. So it's getting around a lot faster than influenza is. And also I think it's a virus that's probably all year round in terms of its seasonality rather than temperate peaks as we currently see with influenza. So that's certainly the case from gizzard and others. That's exactly what we are seeing.
Great. Professor Galleria, you've got a huge population you're representing, and our chairman may wish to comment here as well. But could you sort of reflect on what influenza is doing in different parts of India and how your control measures are operating?
So influenza has been a challenge before COVID, I would say. And I think we are seeing less of influenza, but probably because of the, let's say, COVID appropriate behavior also, masking, physical distancing also would help to some extent as far as other respiratory infections are concerned. And that probably has also led to some decline. I remember reading a paper from Japan where they had very few influenza cases, which could be linked to basically a lot of the physical distancing and masking which was done. But you see, in our country, we've had a challenge because I've been sort of trying to push for the influenza vaccine now for more than 20 years. The challenge that we've had is that physicians themselves in the beginning did not really accept the fact that influenza is a problem in our country. They would say influenza is something which happens in colder climates and we are a tropical country and we don't see influenza that much. We shouldn't really worry about it. Despite this fact that our Indian Council of Medical Research had a flu network and papers from our own center where we looked at seasonality and from National Institute of Virology, Pune, showed that we were seeing influenza in India itself. And this led to a reluctance to really push for the influenza vaccine. In late 90s and early 2000s, we had outbreak of H5N1, which was avian influenza, bird flu, and this caused some concern because in a few states in India, there was a large number of poultry deaths which happened. Luckily, there were no human cases, but we know at that point in time, there were human cases reported from Southeast Asia and from China, and that it has a case fatality of 60% H5N1. And that also caused a lot of concern as far as influenza was concerned. But I think what really woke people up was the H1N1 pandemic in 2009. And that really made people realize that yes, influenza can happen because it led to a significant degree of morbidity and mortality during that pandemic phase. We've been pushing very aggressively for the influenza vaccine to be given to all the high-risk groups in our country. Luckily, now even after the H1N1 pandemic, it's now come into the guidelines even for pregnant women, and it's being aggressively followed. I've really had a lot of discussions, webinars, and talks with our Federation of Obstetrics and Gynecology of India, and they have also now endorsed that the influenza vaccine should be part of the vaccination program for pregnant women, and it has a triple benefit as far as pregnant women is concerned, both for the mother, the fetus, and the newborn. So that has now been accepted, but we are really working hard to get influenza to become the mainstay of treatment for adults, especially in the high-risk group, the elderly, those with comorbidities. It's a multidisciplinary this thing. You have to talk to your nephrologist to get them to agree to give it for patients with chronic kidney disease. You have to talk to the cardiologist. It's easier to get the pulmonologist on board, but we need to get others also on board, and it's happening. And I think COVID will also help to push that. But I'm confident that we will have an aggressive adult vaccination program post-COVID, and that will include influenza also.
Wonderful. Influenza H5N6 is causing trouble in China right now, avian flu, and there are cases and deaths of concern. But picking up on your other point about pregnant women, both North America and Australia routinely recommends both flu and COVID vaccination for pregnant women and newly breastfeeding. Gary, would you like to comment on either of those things, the role of vaccine in pregnant women or H5N6 in China, or a third topic that you think is burning?
Well, it's interesting to see the H5N6s and others arising in China, but this is not news. We see them every year or two, and they're reported quite well through the animal virus network and then the human one. And there's a lot of work going on in monitoring zoonotic infections going to humans. Generally, they're very well controlled. They're not terribly infectious in terms of human-to-human spread. And that's why we basically are on top of the various H types and H5N1 and H5N6 and so on that still exist in the world, but are not really gaining a foothold in terms of person-to-person spread in humans. So that's not surprising. We're all holding our breath, of course, hoping there won't be a particular reassortment or mutation that will allow easy person-to-person spread or efficient person-to-person spread, but so far that's not been the case, which has been very, very fortunate when it comes to those particular viruses. In terms of pregnant women, I think it has to be driven by data, whether we're talking about COVID or influenza. There's enough data with influenza to show how devastating influenza can be to mother and child. It is really, really important that pregnant women get vaccinated to influenza. I'm actually unclear still at this stage what the COVID data says, although most advisory groups seem to suggest that vaccination during pregnancy is okay. But I don't know that that's particularly data-driven, at least not convincing to a conservative ex-regulator like myself anyway.
I think the real-world experience is in tens and hundreds of thousands of pregnant women, but I'm getting a bit of feedback there, hopefully.
If I may, I was also looking at my patients' data about 3,500
but can you mute
patients.
the person in the background for us, please? You've got someone in the background you need to mute.
We have, I've looked at about 3,500 patients' data who have been taking regular influenza vaccine year after year, and who have been given at least one pneumococcal vaccine in the last 5 years. And people who have taken more than 3 years of regular influenza vaccine. When I looked at my data and kind of, we are yet analyzing that data, but my initial feeling is that there is 30% reduction of them getting COVID infection. There may be various factors responsible. People who take regular vaccines are more disciplined, they are more careful. It may also happen that way. Most of my patients are respiratory patients and they are on inhaled therapy. Most of them could be on inhaled steroid as well. It may also be playing a role. But yes, influenza vaccine may have some protective effect against getting COVID infection as well. Sir, you're on mute.
Gonna have to stop you. You've got too much noise in the background. So I'll just move to Professor Galeria. You're double sounding us, so we can't deal with that. Professor Galeria, please go on mute. Professor Galeria, can you talk to nasal steroids and other approaches?
Okay, so the issue is that as Dr. Vora mentioned, that there is a lot of, since both of us are pulmonologists, we see a lot of patients with allergic rhinitis and asthma, and we do tend to use a lot of nasal inhaled steroids, especially as a part of allergic rhinitis along with antihistaminics. And that continues because that treatment strategy is something that patients would need. And that is part of our treatment protocol as far as allergic rhinitis is concerned. So I'm not sure in what context the question was asked, because there was a lot of background noise which was happening and one couldn't really hear the question per se. You're currently mute.
There's been a question in the chat about what happens in the next 12 months and boosters and the like. One of the people discussing in the chat has asked about protein vaccines. Is that something that Paul Griffin would like to speak to? You've been doing trials on various different vaccines. You can talk about protein and other vaccines and boosters as they happen over the next year.
Look, I think with that question, we've been extremely fortunate to see the successes of the vaccines that we already have available, but they're not perfect. And I think there's a number of properties we'd like to improve, particularly transmission blocking, for example, stability, longevity, route of administration, for example. So there are lots of properties we'd like to improve on. And I think we'll have second or third generation vaccines that will improve on some of those. They may not replace the current vaccines, but may be used in a complementary fashion. And some of these may be able to be given intradermally or intranasally, for example. And there's lots of promising, I think there's around 6 or 7 intranasal candidates that are looking quite promising. And we're yet to, I think, realize the potential of the protein or subunit-based vaccines as well, which I think will have some complementary properties to the viral vector and the mRNA vaccines that we're using most commonly. So I believe there's 99 vaccines that have entered clinical trials. So there's a lot more to come, and I'm really excited to see what additional properties they'll give us when we use those complementary to the current vaccines.
Gary, you added something in the chat. Over to you.
I just added, I thought that boosters would be essential. It's a question from Sheldon. I think, I think given the way and the rate that coronaviruses are evolving, and the GISAID database is pretty clear. I mean, we've seen some 28 variants really come and go in less than a year and a half, 3 or 4 of them quite serious and very efficient person to person. So we can expect more of them. And so boosters, I think, will be essential as antibody wanes after 6 to 9 months, depending on what paper you read. And also the virus will be changing. So like influenza, we're going to need to update it, in my view anyway, and take a booster once a year, perhaps every 9 months, to ensure that variants don't spread and the vaccines don't become as effective as they might be. And I mean, as Paul alluded to, the protein vaccines, I think will be the ones that would be very effective in this area. Of course, we do need to make them quickly, and from the point of view of speed, mRNA would certainly be the fastest in terms of updating a particular variant and making a new vaccine very, very quickly. So it might then mean a multi-pronged approach. But as I alluded to before, there's actually no data on how many mRNA vaccines a human being can have. And that's a question from a regulatory point of view that's yet to be answered. We know they can have 2 and possibly 3, but we actually haven't even got animal studies on that are definitive. So that's still a question mark.
I'm going to go to Sheldon, even if it's only briefly, and then over to Professor Galeria again. So I want to hear more about the vaccines being made and developed and implemented in India. So Sheldon, you wanted some brief comments before we go over to Professor Galeria?
Oh, I think what he's got to say is a lot more interesting than what I've got to say about vaccines.
Okay. All right, Professor Galeria.
Okay, thank you very much. So what we started off was trials which were done. First, of course, the bridging studies for the AstraZeneca vaccine, which is now being produced by the Serum Institute of India. We also have the Bharat Biotech, which is now a live attenuated whole viron vaccine, which is also available, and that shows an efficacy of around 78 to 80%. They've all been looked at as far as the Delta variant is concerned, and although the efficacy has come down, it's still significant. It's in the 60s, but does give good protection against severe disease and hospitalization. And I think most vaccines are actually providing good protection against hospitalization and severe disease. They're not really that good in terms of preventing infection per se. And that is why we still continue to see increasing cases in various parts of the world, although we're not seeing increased hospitalization or mortality. Having said that, I do agree that we will have to have a booster dose. The challenge is with so many vaccine candidates coming out, what is the ideal mix and match as far as the booster is concerned? Should it be, as has been in some trials, that you give the AstraZeneca and then you can use the mRNA vaccine, the Pfizer, and you may have some degree of reactogenicity, but you have better immune response? Or should we look at the other vaccine candidates? I don't think we have an answer, and we need data in terms of what mix and match is going to be good in the long run. There are some studies which have been initiated in India also trying to look at the mix and match as far as vaccines are concerned. And like I mentioned, they are also looking at new vaccine candidates. What is exciting is that we have now approval for a DNA vaccine, which again, like the mRNA vaccine, can be made much more quickly, and is a novel vaccine, and it's used a different mechanism as far as the delivery is concerned. It has an intradermal delivery device, and you need 3 doses rather than 2 doses as far as that is concerned. The Johnson & Johnson vaccine has also tied up with a company in India for manufacturing, and that should also be available in India and for global export also. So I think in the coming few months, we would have a number of vaccine candidates being produced in India, and we need to really then try and see what is the best combination for our population, and do we need to give different vaccines for different groups, or say for children, do you need something different? Do you need something different which could be more effective for the elderly or those who are immunosuppressed, where the immune response may be less? And how many doses should you give for those whose immunity is low? Should we go on, as some data suggest, that they should upfront get 3 doses rather than 2 doses as to build up their antibodies? So I think these are questions that we are still sort of trying to answer and develop guidelines as far as these are concerned.
Professor Galleria, before we give anyone 3 doses, can you tell me how much production is happening and being used in India, and how much is being exported to the rest of the world by the Serum Institute? We sort of got a bit lost with Serum Institute making a lot of hundreds of millions and giving them to the rest of the world, and then having to stop because India got in trouble. What's the situation now?
So this is quite a politically loaded question, if I put it that way, because this has got a lot to do with the government policy. Because in the early stage, India had started exporting vaccines to neighboring countries, the Serum Institute, and were also giving it as far as the COVAX program of the WHO was concerned. This created then a lot of local political issues that we are not vaccinating our own population and giving it to others. And I think that is not correct. We need to really understand that we need to vaccinate the world if we need to come out of this pandemic rather than regions. Because in any area, if the virus continues to multiply, it will mutate and develop new variants which may cause the vaccine to lose their efficacy. So I think because of the fact that it has become sort of more politically driven rather than science driven, there is less amount of vaccine which are going to our neighboring countries. There was a vaccine which was being given to Bangladesh, Nepal, Sri Lanka, the other countries. That has really come down significantly. One is very hopeful that by next month, we should again be able to start giving out vaccines, because now new plants, both Bharat Biotech and Serum Institute have set up their new plants and have also signed MOUs with other vaccine producers so that these vaccines can be produced by other manufacturers also in India. So that will really help in increasing the number of doses that are available. So one is hopeful that by next month, we should be able to again resume providing vaccines to other parts of the world.
Wonderful. Professor Sheldon Campbell has a comment or a question.
I you know, from my country's perspective, we can't talk about politics driving policy instead of science. So I got nothing to say about that. But from a diagnostics perspective, I'm intrigued by the possibility that we might have a serological method that could help inform some of these decisions about vaccines. So I'd like to ask the vaccine experts to tell me what you think about, can we correlate antibody levels with protection to help to either customize when we do boosters in individual patients or in populations? Is there any way the diagnostics world can help inform those decisions? Or is the protection and antibody levels, which is the only thing we can practically measure on a large scale anyway, a little bit too attenuated in their associations?
Can I just
I'm going to throw that
question?
Go on.
I just wanted to ask that from the experts that, you know, we would it would not be ethical to do a randomized controlled trial using a placebo. We will have to do inferiority trials by and large, or we will have to develop a correlate of protection. What is a correlate of protection in terms of the antibody level, or whether you're looking at cell-mediated immunity? No one really knows that. If you say I get patients who ring up and tell me my antibody level has fallen. Should I take another booster dose? Should I take the third dose? I don't know what is the antibody level which I can say is now giving you a lack of protection as compared to giving you good protection. Do we have some data on this, and what is the science moving forward? Maybe others could answer that. I really don't have an answer to that.
Well, we've posed really two good questions that are related, and I'm sure Paul Griffin will have an answer, and then Gary Grohman, and then I'll have a go as well. Paul.
Look, it's a tricky question. It would be great to have a simple test we could use to know who needs to be boosted and when. I don't think we have that yet, though. I think the correlation with antibodies is perhaps not quite strong enough to use that solely to make the decision. In some studies, my understanding is that 20 or 30% will be seen to be non-responding if we measure antibodies alone, whereas we think that perhaps the cell-mediated immunity may still be robust enough to provide sufficient protection. And in countries like ours, we simply don't have access to a lot of serology through our routine diagnostic labs to make that remotely feasible at this point in time. So I think we're going to have more of a broad campaign than doing it more specifically.
Gary.
Yes. Look, I think this has been one of the flaws since this whole thing started, is that we really don't have proper correlative protection. We've taken a guess at antibody to the spike protein and some others, but there is no correlative protection. And normally, I would have thought for a virus like this, you would hopefully get a correlate such as IgA, an antibody produced at the site of infection. Now, the work really hasn't been done. I don't think it's anybody's fault. It's been, in part the speed, and we've made this assumption that a particular antibody marker means protection, but it's absolutely an assumption. It's not a fact, and we're using it, which has been one of the flaws in the whole thing, in my view, an interesting flaw, but nevertheless, one of those things that could come back to bite us. And I don't know how other regulatory bodies are going to think around the world, but it's one of those elephants in the room. What is the correlative protection? And we're actually using a surrogate to make decisions, policy, scientific decisions, and so on. So this is something that really does need study and needs to be cleared up quickly.
It's perhaps an inevitable flaw given the pace of things, but we need to keep in mind that we are operating on assumptions rather than data in that realm.
And I'll just give the optimistic view as a pediatrician who did my first vaccine study more than 30 years ago on Hib, Haemophilus influenza type B, and we were establishing way back then what the threshold of antibody levels were for shorter and longer term protection. And that was underlined through the 1990s. So I do think it's doable. And I do also think from an epidemiological point of view, rather than immunological, that we've gathered so much data on prevention through efficacy studies and effectiveness, but let's just focus on the efficacy studies, that the antibody levels that we've measured and the T-cell responses that we've measured can be used to get some kind of reasonable correlate of protection, because we've got that in the trials. We've got antibodies and we've got disease occurrence or not. So I can't see us not going more than another year without better answers to these questions. And I'm not going to provide them, but someone will, in my optimistic opinion. Okay, we've got just a few minutes to go. I haven't seen too many questions in the chat, so please be encouraged to throw some more questions in. I'd like to turn back to Professor Galeria, because the experience is just so deep and huge that I'd like to know what you think the world should know about what you've learned from India and how we could better manage this problem based on your experience and based on all the reading and the clinical work that you've done.
Thank you. So I think there are 2 or 3 things that I would like to point out. One, of course, I think like we've invested a lot on vaccine and vaccine development, we need to invest on antivirals also. We still don't have a good antiviral drug. We have monoclonals, but from me as a clinician, I would like to have a good antiviral drug which could be useful as far as COVID is concerned. What we have are repurposed drugs. Even remdesivir is a repurposed drug where efficacy is still doubtful. And that holds true for various other drugs which have been used or are available in the Indian market. We have ivermectin, favipiravir, so many others which are being used. But there is really no concrete data, and we need to really invest a lot as far as developing a good antiviral is concerned. The second thing I think is that this is a global problem, and therefore we need to really develop a global aggressive surveillance network so that we are able to pick up not only a hotspot, but new variants which may emerge from one area, and we're able to take action aggressively rather than allowing them to really spread across the world, as has happened both with the Alpha variant and then subsequently with the Delta variant. The surges that we're seeing are basically because these variants have really spread aggressively before aggressive surveillance or containment strategies could be put in place. And like we've said, that we need to really develop strategies on how do we move forward as far as vaccine research is concerned and the booster dose is concerned. So these are the experiences that we've had. I personally feel that from clinical point of view, we need to try and be as much ahead of the virus that we can be. Currently, more often than not, we're chasing the virus rather than being ahead of it.
Seeing as we're chasing the virus, and seeing as India indicated that Delta Plus was a variant of concern, but the WHO hasn't yet concurred, would you like to comment on whether Delta Plus remains a variant of concern, or whether there's now an Omega virus that we should all be scared of in India?
So the data that we have, although it's still emerging, doesn't really suggest that it has moved from a variant of interest to a variant of concern. But we're still trying to get more data in terms of mortality, in terms of it being more infectious. But whatever data that I could look at, I have not really seen that wherever the Delta Plus has been reported in small pockets, there has been more hospitalizations, or there has been a huge surge in the number of cases. So these are early days, and we must understand that the virus continues to mutate and change. And that's why we need to be very vigilant and need to monitor it more closely, because it could lead to some new mutation happening, which could really change the whole story.
Gary, you've been following GISAID very closely. Is there anything you'd like to add?
Look, I mean, in my view, it's very, very hard to predict what's going to happen in the future, particularly in relation to severity. But we do know that there will be new variants coming. So I'm kind of reluctant to make any predictions at this point. But generally, for viral infections overall, the tendency is to less severity, less mortality over time, but becoming more human adapted. So that tends to be the case in general. I think you use the word catastrophize. I think what happens is that people in the media and even scientists politicians particularly tend to catastrophize everything, and we must be careful not to do that and really stick to the data. And it's not an easy thing to do, as you know, we're surrounded by people who like to catastrophize things, and you know, the end of the world is nigh, and so on. There is certainly there's certainly good scientific data out there that we can focus on and have confidence in. And a lot of it does come from GISAID, in fact. Over.
Sheldon, I'm going to quickly change tack because we've got about 2 to 3 minutes left. I'm intrigued by diagnostics when we've got all these co-infections, whether they're mycobacterial, whether they're fungal. What are the key diagnostic tests that we should be doing in severe cases of COVID when we don't know what the co-infection might be?
Oh, I think that falling back on good clinical judgment is the only place to go. We know that in profoundly immunocompromised patients, there are many possibilities, and I think that severely ill COVID patients, particularly those that have been treated with steroids or other immunomodulatories, fall into that category. So testing more broadly rather than narrowly is probably the way to go. I'm not sure there's an efficient guidance there because of the nature of those patients.
Can you hear me? If there's a question in the chat that Gary might address about next generation sequencing to predict future viral variants, some kind of machine learning perhaps. Gary, can you hear me, and can you address that question before we run out of time?
Well, it's the new science, and I think, yes, that is going to help us a lot. And the more we get data, particularly deep sequencing kind of data, and the more of it we have around the world, then certainly that's a possibility, and that's where a lot of people are heading towards. And I think that will be incredibly useful in time.
Gary, while you're there, there's another question about T-cell immunity measures to correlate with protection. Yeah. Would you like to comment on that before we run out of time?
Well, just very quickly, the whole issue of correlates also involves, of course, the other half of the immune system, so to speak. It's not just about antibodies. And again, we really don't have any information that we can rely on to decide that there might be a particular correlate of protection. At the moment, again, we just have immunological reactions and biomarkers at this stage.
Okay, lots to learn. Professor Galleria, I'll give you the final word. You're the one with the experience. What do you think we should be doing, and what's the way forward from here?
I think the way forward is to really continue to interact and really learn from each other. That is one. And secondly, keep a close watch on how things are evolving, be aggressive. I think we have to move just not from only testing, but to really do genome sequencing to pick up the new variants. And like I mentioned, work on trying to develop and support research on good antivirals, because we still need a good antiviral drug as far as treating COVID-19 is concerned.
Brilliant, thank you. Can I thank Professor Galleria, Professor Groman, Professor Griffin? Everyone's got a G except for Professor Campbell. So it's been great to interact with you all. It's been a super session, and I thank you all, and I hope the audience have learned something. And I wish you all well and stay well. Cheers. Bye.
Thank you.
Thank you.
Thanks for your moderation. It's excellent.
Okay.